Thank you for joining us today. It's now my pleasure to introduce our speakers, Raja Majed, PhD, Technical Sales Specialist from Refine, and Quentin Bazot, PhD, Head of Innovation and Process Development from ABL.
Hi everyone, thank you for your time and for being here today. I am Rasha. I'm a technical sales specialist at Refine, a company that commercializes mass photometry. So, as you might be aware, one of the biggest challenges in the gene therapy industry is the level or the high level of empty particles or empty AAVs that can be produced, because those high levels of empty particles can pose several risks, from low transduction efficiency to an increase in the immune system to an increase in general of the cost of goods produced. So that high level of empty particle, it's due to the fact that there is a gap in a good quantitative accessible analytical tool that allows a characterization of the AAV samples at early stages. So when you were thinking about developing a new technology, we thought the ideal solution would be a system that is accurate for empty full ratio characterization that gives you a result within minutes, that should be serotype-agnostic, requiring a very low amount of sample in terms of volume and particle titer. It should as well provide an idea and quantify partially filled capsid, and it should be as well applicable to all or to a different purification stages. So all of those points we took into consideration when we have developed the SAMOX MP, the mass photometer dedicated for AAV analysis. So the SAMOX MP, it is based on our core technology, which is mass photometry. It is a type of microscopy where you have as a sample carrier a glass surface. It's a standard microscopy cover slip where the end user will come pipette their AAV in solution in your buffer of choice without any need for labeling or extra treatment. Then we shine a laser in that direction. Part of the laser will be reflected by the glass surface and collected by the objective. And since the AAVs, the resolution, they diffuse around to come close to the glass surface where they bind and they scattered light. What we quantify actually is the interference between the scattered and the reflected light, resulting in a contrast like you see here. The intensity of that contrast scales linearly with the molecular mass of the particle itself. So our technology, it's purely based on molecular mass, which is ideal in AAV context, because the difference between an empty and full AAVs is fully based on molecular mass. What you see here, it is a live image. It's a video that we record for a minute of time, and after that we took that acquired data to our analysis software, where you have an output or a result like so, where you have on the x-axis your molecular mass, and then on the y-axis your single particle count, because as I mentioned before, the SAMOX or mass photometry, it is a single molecule approach where each AAV is quantified. So based on our resolution on the system and based on the mass shifts between the different AAV population, we would able to distinguish and to separate between the empty population, the full populations, the intermediate or partially filled AAVs that usually sit in between, and if you have aggregates or overfilled capsid, they should as well we be able to separate them and quantify them as well. The SAMOX MP, which is the system that I highlighted before, comes more in a manual fashion, where it's manually operated by the end user, or it can come in an automated fashion where the user have ability to run 24 measurements operator free, with fully automated without any like user interference. Both the SAMOX and manual or automated provides an accurate and fast empty full AAV characterization of any serotype within minutes, requiring very low amount of samples. So usually we need 10 microliters from E10 10 to the 11 particle per ml. It is very simple and very easy to use, and we have as well a very streamlined data analysis software, which is make the whole user interface very easy and friendly as well. I will give now the stage to Quentin. Quentin will be diving more deeper or more in details into the challenges faced in AAV manufacturing and how they have implemented the SAMUX for AAV characterization and how actually they optimize their downstream processes by the SAMUX MP.
Hi everyone, my name is Quentin Bazot. I am leading the innovation and process development team at ABL. So ABL, we are a CDMO specialized in viral vector production, and we have two sites in France, one in Strasbourg and one in Lyon, where I'm based. And today it's actually my pleasure to be here and to be talking about the mass photometry technology and how we are using it to accelerate AAV process development and characterization. First of all, I wanted to go back on one of the main challenges in AAV manufacturing. So here in this slide, you have a schematic of a classical AAV process, starting with the upstream. So that's the cell culture stage, and that's where AAV is going to be produced. Then you have the downstream part, and that's where AAV is going to be purified. And so one of the main challenges in AAV manufacturing is that actually most of what we are going to produce after the upstream will be empty. So we will have empty capsids, partially filled capsid, and our AAV full capsid, which are our particles of interest. Then it's down to the downstream to obviously remove impurities and enrich for our full capsid. So this full empty ratio is a very hot topic in the AAV field, and that's a very, very important metric that we should characterize as soon as possible, as early as possible in a process, AAV process, especially when we're doing AAV process development. And then we should also follow this metric along the process. And I have to say that a few years back, it was not impossible, but very tricky to follow this metric in an accurate way and a quick way. And really here, the mass photometry helped us a lot, and it changed the way we are following the empty-full ratio during AAV process development and characterization. And I hope that today I'll be able to convince you that the mass photometry is actually a game changer in the AAV field. So my talk today will be divided into three parts. First of all, I'll be talking about the assessment of AAV standards. So that's one of the first things that we did when we assessed and acquired the mass photometry technology. Then I'll be talking about early AAV characterization. And then last but not least, I'll be talking about the optimization of the AAV polishing step. So if we start with the standards. So we had in our QC lab some AAV full standards. Those standards were and are commercially available. And we had already quite a lot of data on those standards thanks to the supplier. The supplier had performed two orthogonal methods, which were the SLS-UV and the CDMS, giving us already a nice estimation of what we should expect in terms of percentage of full from those standards. So we use those standards in the SAMUX MP, and this is a first example with AAV5. It worked well. We had two peaks, empty peak and full peak, and it worked well not only for AAV5, but for the other serotypes tested, which were AAV6, 2, 8, and 9. Then obviously, the big question was, how does the data obtained with the SAMUX MP compare with the orthogonal method data from the supplier, and especially the CDMS? Well, everything is in the table in blue, and as you can see, it's really similar to the CDMS and also the SLS-UV. If we take, for example, AAV6, when we're expecting to have 92% or 93% of full with the SLS-UV and CDMS respectively, we got from the SAMUX MP 90%. So it's very similar to the CDMS data and SLS-UV data. Then we decided to look at another interesting feature from the SAMUX MP. So the SAMUX MP being the equipment, which is a mass photometer. And this feature is the AAV titer estimation. So first of all, we looked at the viral particle titer estimation that we got from the SAMUX MP for all our standards. And we compared them with the ELISA data that we had from the supplier. And everything is in this table here and is expressed in VP per ml. And well, as you can see, we are just a little bit less with the SAMU-XMP compared to the ELISA. And in average, it's 1.5 times less, which is quite close. Then if we focus on the full peak, we can actually extract the phylogenomic titer in VG per ml. So we did that for all the standards, and then we did compare them with the qPCR data that we had from the supplier. And here, we're actually very happily surprised to see that they were the same as expected. If we take the example of AAV5, for instance, when we're expecting 2.5 E12 VG per ml, we had actually 2.66 E12 with the SAMUX. And for AAV8, when we're expecting 3.5 E12, we had 3.6 E12 VG per ml with the SAMUX. So very, very similar. So in conclusion, this AAV titer estimation works quite well and is a very interesting feature, especially when you're doing process developments, to have a rough idea of the concentration that you have in your sample. Then let's talk about early AAV characterization. First of all, I need to mention that, at least in our hand, in order to use the mass photometry to characterize our full empty ratio, we need to have a sample that is a little bit purified. And for us, it's after the capture step purification, which is here an affinity chromatography. We try to use lysates or clarified bulk, and it doesn't work because there's still a lot of contaminants. So here in this example, we have two AAV9 production with the same system. So by system, I mean the same plasmids, the same transfection reagent, and the same cell line. The only difference is the transfection parameters. So here we ended up with two phylogenomic titer. For the first one in orange, we had around 2 E10 VG per ml. And for the second one in blue, we had a log higher, 2 to 3 E11 VG per ml. Then if we look at the percentage of full AAV expected at the harvest, because here everything was at the harvest step, we were expecting 34% and 9% of full AAV if we do the ratio PCR-ELISA. We all know that the ratio PCR-ELISA is not the best accurate way to assess the percentage of full AAV, but you'll see that sometimes it's quite useful. So then we characterize those production using the SAMU-XMP, and when we're expecting to have 34% of full AAV, we got actually from the SAMU-XMP 40% of full, which is very close. Then when we're expecting 9% of full AAV with the ratio PCR-ELISA at the harvest, we actually found out that it was 9.4% with the SAMU-XMP after the affinity resin. So once again, very, very close. Then now if we focus on the orange box here, you will see that, well, you can see your empty peak, your full peak, and you can see also here some signal in the low molecular weights. So those are impurities, contaminants, and they can be found in this experiment, but not in the blue one. And the reason is here, the two affinity capture steps were done independently using different process parameters, but also different buffer elution buffer. And this is key. And we're using also the mass photometry in order to assess the purity of our samples that comes out of the affinity resin. But here really the point of this slide was to show that actually the full empty ratio that we found with the SAMU-XMP correlates very well with the PCR-ELISA ratio that we have at the harvest step. However, can we always trust this ratio PCR-ELISA? You will see that it's not that easy. And the simple answer is no. So this is an AAV2 production, this time using different plasmid system A and plasmid system B. The very important point is that they both had in common the GY plasmid. The exact same GY plasmid was used. So we ended up with two different titer. So for plasmid system A, around 2 E10 VG per ml for AAV2, and for plasmid system B, a log higher, around 4 to 5 E11 VG per ml, which is great. Then when we characterize them on the SAMU-XMP, when we're expecting to have 18% of full AAV, if we believe the PCR-ELISA ratio, the SAMU-XMP confirmed that it was actually 20%, which is once again very similar. With the plasmid system B, when we're expecting to have 90% of full, we got 17% of full, which is once again very similar. However, if we look more carefully on the plasmid system B graph, you might have seen by now that there's something that is not right. And you might see it even better if I combine the two graphs together. Because not only you can see your empty and full peak, but with the plasmid system B, you have a lot of signal between those peaks. And here are your partially filled capsids. And that's one of the beauty with the SAM-XMP is that not only you can see your empty and full peak, but you can also see the partially filled capsid and the overfilled if there's any overfilled. So clearly here, early AAV characterization is essential for process development. Otherwise, well, you are blind if you just believe the PCR and ELISA ratio. Then we wanted to see if we could confirm the size, the mass of the encapsulated genomic cargo with the SAM-XMP. So here, once again, two case studies. The first one with AAV2 that has a genomic cargo size of 4.4 kb. So we can translate this size in kb into molecular mass. So the expected mass was 1,417 kilodalton. And here what we did is we took the average mass of the empty peak, the average mass of the full peak, and then you can easily calculate the average mass of the cargo. And here, actually, we found 1,353 kilodalton, which is very similar to the expected mass, with a percentage of error less than 5%. Then on the second case study, here it was AAV9, with a size of the genomic cargo smaller, 2.2 kb. And here, after affinity capture resin, we had actually 3 peaks. So the first peak is the empty peak, and then we had 2 other peaks over there. And by looking at the mass of those peaks, we actually found out that this peak one was our AAVs containing 1 copy of the genome, whereas the second peak here are our AAVs containing 2 copies. And once again, when you look at the table and when you compare the expected mass compared to the calculated mass with the SAM-XMP, you'll see that it's once again very similar. So the answer is yes, we can assess and confirm the expected mass size of the encapsulated genomic cargo with the SAM-XMP. Then I wanted to talk about the polishing step. So the polishing step is maybe the most technical, challenging step in AAV processes, and that's the step where you want to enrich for your full AAV. So here, this is an example with AAV9 polishing. And this was an experiment that we performed with a feed that we had no idea about. So what we did is we used our standard anion exchange chromatography protocol, and we ended up with this chromatogram. So the first thing to do is to try to read the chromatogram. How do we do that? Because protein absorbs in the 280 nanometer wavelength and nucleic acid in the 260 nanometer wavelengths, you can actually look at the ratio between 280 and 260. And when you have a higher signal for 260 nanometer, it does generally look good. And it means that you might have enriched for your full AAV. So here in this chromatogram, well, this peak would look good. So before the mass photometry, what we would have done is would have taken the fraction of interest, send that to our QC team, wait for the results, then analyze the results. And then, well, if we're happy with the experiment, that's great. We're doing it again and we confirm it. Or if we're not happy, well, we're doing the experiment once again. And this would take a lot of time. And now really the mass photometry changed the way we are optimizing the polishing step. What we did here is we actually took all the fractions and we analyzed them with the SAM-XMP. So it took us maybe 2 hours, but in 2 hours, you have a clear picture of what happened during the polishing step. So what I wanted to show you today is obviously we looked at the load and we also looked at the flow-through samples because we had some signal in the flow-through. So everything that didn't bind to the column. So as you can see, we have our loads, empty peak, full peak, and in the flow-through, we actually lost a little bit of empty AAV, which is good. Then if we focus on the elution, which is an elution, a gradient elution, we had a lot of peaks, And what we did is we obviously looked at different fraction. We took some fraction of this huge peak over there. And as you can see, there's a tiny peak before the big peak, which looks to be empty. And actually, this is the case. The fraction B9 to C1 are mainly empty. And that's quite interesting because the C1 fraction here, and if you look at the chromatogram, this C1 fraction is right in the beginning of, it's over there, right at the beginning of the peak. So you might have thought that it's full, where it's mainly empty. Then the fraction C3 to C9 were the one where we actually enriched for our full AAV. We also looked at the other peaks of the chromatogram. So here, D4, D8, and E6. D4 is mainly empty. D8, well, okay, we are losing a little bit of full AAV. And E6 on the chromatogram, that was quite weird. We had a huge signal for the 260 nanometer wavelength, but actually it's empty. So actually what we did here with this experiment, we didn't send anything to the QC team. We came back in front of the chromatogram and we walked around the process parameters. So we've looked obviously at the conductivity with the aim of developing a two-step gradient solution where we could separate the empty fraction here from the full fraction. We had also a target of removing empty particles during the load. So actually what we did is we performed another chromatographic run on the same day. And we ended up with this chromatogram, this experiment. So we've pulled out the fraction. And so if we analyze that through the Simux MP, so this is our load. So we started with 15.7% of full. Then in the flow through, actually, yes, we got rid of some empty particle, which is good. And in the pull fraction of the full, so we actually pulled all the fraction here, all the peak, we ended up with 78.4% of full, which is quite nice because it's 5 times enrichment for the polishing step. So in conclusion, the mass photometry technology is, I do believe, a game changer in the AAV process development and characterization because it has a very quick turnaround result, less than 2 minutes per sample. Very small sample volume is needed. It works with different buffer, different matrix. It leads to robust results, and it's very simple to use. And really, the use of mass photometry decreased drastically development time and the cost of analytics associated to the development studies. So thank you very much for your attention. I am very happy to take any question at the end of the webinar. And I am now giving back the stage to Rasha, who is going to wrap up the webinar. Thank you very much.
Quentin summarized very, very well the advantage of the Simux for AAV characterization. I just wanted to give you a few examples how we compare the Simux MP compares to the gold standard in the field, such as cryo-GM and AUC. So this type of results were done in collaboration with Catapult and Catapult and Pharmaron in the UK, where they have compared the Simux MP or the results of the full proportion coming from the Simux MP to cryo-GM and AUC. And as you can see here, the results compares very, very well. They correlates very, very well. However, as mentioned in Quentin's talk, Simux, it's very, very easy to use. It's fast and requires very low amount of samples compared to other more expensive techniques such as cryo-GM and AUC. Before wrapping up the webinar, I would like to highlight as well some very exciting news that we have launched the 21 CFR Part 11, which allows the Simux to be installed in a GMP compliant environment because previously the Simux we offered the Simux in more R&D, process development and analytical development platforms. However, now with the GMP compliant software, the Simux can be implemented in QC and release assays. The refined GMP or our GMP offering includes 3 parts. So we have the refined manage MP. It is a platform that allows you to manage user authentication and access control. The refined manage MP allows as well an easy way to visualize and to export audit trails. and as well it allows you to define system configuration as well. We have the Acquire MP, which is our software for data acquisition, and Acquire MP as well is a platform that allows you to operate the mass photometry and as well to acquire data. We have the Refine Evaluate MPS, which is which is our data, like GMP data analysis software. It allows an easy way to determine AAV ratios for full, empty, and partially filled as well. And as well, it allows to export data with electronic signature. We can, of course, discuss your specific GMP requires and needs and dive a bit deeper into that. So please feel free to reach out to us, and we are happy to take that a bit further. And with that, I would like to wrap up the talk, and Conta and I will be very happy to answer any question you might have. Thank you again for your time and for being here today.